Thanks for those very kind words, Peter. So this project has been about market access treatments for horticultural commodities, not only for those pests that are in country, the market access pests that are here already, but also for those that may arrive and become market access issues. This is the team that worked on the project from a dozen organisations, 3 countries, and as you read through the list, I'll tell you that we had a lot of laughs, we had a lot of heated discussions, we had a few arguments, but all in all, we had a pretty good time. So quarantine pests, like these, are a major disruption to horticultural trade. And in order to export to some markets, most markets here in Australia and for some markets in New Zealand, you need quarantine treatments. So we took this opportunity in this project to review 40 years of post-harvest disinfestation research and identify the priorities for market access research, collate data to support negotiation of new protocols. So this was the first task, to create this review document. All 192 pages of it, research from 40 years of disinfestation technologies. We looked at the technologies, we looked at all the protocols generated from the data, we looked at all the markets, the volume of fruit and vegetables going to each market, so that we could see which protocols were being used and which protocols weren't being used. We then had stakeholder workshops in Australia and New Zealand, and we presented the findings from the review, and we talked about their market access issues with them. And we prioritised market access research for each industry with each industry. And those priorities can be found in the stakeholder reports and those priority tables the industries are using to fund R&D in the market access space. At those workshops, we also prioritised the research for phase 2 of the PBCRC project. In the Australian workshop, they wanted to develop new, shorter disinfestation treatments for air freight, and in the New Zealand industries were really keen on developing pre-approved quarantine treatments for fruit flies before they arrive in New Zealand. So the Australian industries wanted shorter treatment for air freight because they wanted to get their commodities to market faster and target market opportunities like Chinese New Year. So some of the cold treatments were quite lengthy, and so John Goldine and his team worked on cold and combined it with controlled atmosphere, ethyl formate, low pressure, and found that a controlled atmosphere cold treatment of 3 degrees for 3 days using 1% oxygen, 95% CO2, followed by 5 days at 3 degrees in air, seemed to be the best treatment that they'd tested. It was a total 8-day treatment, and they did a large-scale trial on Queensland fruit fly, got a really high level of efficacy, unfortunately 3 surviving pupae. So I think the next step for this project is to go back and change one of the parameters, perhaps go down to 2 degrees, so they can get those 0 survivors in the large-scale trial. The Kiwis and some of the Australian industries wanted to develop treatments before fruit flies arrive because, of course, in a fruit fly response, you get these no export zones, and those no export zones are dependent on the market. These is the no export zones that were put in place during the Queensland fruit fly eradication in 2015 in Auckland. Now, had those fruit fly been found in these 2 areas where most of kiwi fruit and apples are produced and exported, then there would have been no exports until a treatment had been negotiated. And of course, it takes time to develop treatment protocols. And that was the case in 1995 when Oriental fruit fly was detected in far north Queensland. Industries lost their markets until treatment protocols were developed. So let's do it. Let's start developing these cold treatments before they arrive for a whole range of fruit fly species. So we focused in on Queensland, midfly, oriental and melon, all of them threats to New Zealand, the last two being threats to Australia. We focused in on apples, a high value crop in New Zealand, potentially high value crop in Australia, and Zespri funded the kiwi fruit component. The commercial cold storage sits somewhere between 0 and 2 for these crops. Temperature fluctuations and shippings can go up to 3, so we wanted to investigate a quarantine treatment of 3 degrees, and the closest market for these crops, it takes about 19 days to get there. So we had a project workshop about 12 months ago, and we looked at the existing cold treatment. From that massive review, we had gathered reports and papers on cold treatment research, and we asked, could we use the data in these reports and papers to provide evidence for the comparison of the cold tolerance of these 4 species? But in the studies, the researchers had used different methods of infestation depending on what the host status of the fruit was, whether it was a good host or a bad host, and that was so that they could get high numbers in the untreated control to compare with the treatments. They used different assessment methods, whether they were looking for larvae in the fruit, pupae emerging from the fruit, or emerged adults. They also used different statistical analysis, and they reported on different lethal times, so it was really difficult to compare species and life stage tolerance of these trials that had already taken place. So we had this very, very clever student, Andre Balve, and under the watchful eye of John Maindonald, aka Gandalf, we created this cold data package where we requested raw data from researchers. The package had templates for adding new data consistently. We could look at different data transformations, fit different models, and get these really cool graphical outputs. So this is the lethal times for 99% mortality of Queensland fruit fly on various commodities. And the first thing you can see is that cold tolerances of Queensland fruit fly can be quite different on different commodities. Now within each commodity, the top being nectarine, plum, grape, each line is a different life stage. So the second thing you'll notice is that the third instar larvae, the purple line, always seem to come out ahead of the other life stages. Then we could do some cool comparisons. We could get the Queensland fruit fly work on at 2 and 3 degree on grapefruit, compare it with the mid-fly work at 2 and 3 degrees. And you can see that mid-fly is more tolerant than Q-fly on grapefruit. But if you look at the 2 degree, compare 2 and 3 degrees within each species, there wasn't that much difference. The thing was, with the cold data package, we didn't really get enough information on melon fly and oriental fly, but we did have some papers with summarised results on a range of commodities, which kind of suggested that mid-flies more tolerant or equal to melon fly, which is more tolerant or equal to Q-fly, which is more tolerant or equal to oriental fly. So we thought, bugger it, we're going to have to start testing these things on kiwi fruit and apples. But the cold data package had provided us a really useful tool for a repository for cold data information, and we could compare across a range of studies. We could see what was missing. We could see we required more data on melon and oriental fruit fly, and there was very little data on kiwi fruit and apples at 3. So we started the process for developing quarantine cold treatment for fruit flies on apples and kiwi fruit, thinking, let's see how far we can get in the next, by this time, 10 months. So we looked at host testing. We still weren't sure about melon fly in terms of its host status on apples and kiwi fruit. We were wanting to work out the most tolerant species and life stage so that after the project we could do large-scale confirmation tests. So, apples and kiwi fruit are not good hosts for these fruit flies. We sent apples and kiwi fruit over to Hilo, Hawaii, where Peter Follett exposed them to each fruit to 25 gravid females of medfly or oriental fruit fly or melon fly, and he compared that with papaya. And on that graph you can see that he used punctured and unpunctured fruit, and there were heaps of eggs laid into the papaya. On the melon fly there was hardly any eggs laid on the unpunctured fruit, and so these flies were not good. These fruit were not good hosts for these flies. Meanwhile, Samuel Brown made best friends with Peter Leach and his team in Cairns, and he was looking at the larval development of Queensland fruit fly in a dwarf banana, jazz apple, gold kiwi fruit and green kiwi fruit. And so he was injecting 100 eggs into each of these fruit, and he was getting 53 pupae coming out in the bananas, 2 coming out in apples, 6 and 6 in the kiwi fruit. So what did this mean? This meant that the project got bigger than Ben-Hur. This is Sam and Dom being Ben-Hur and infesting thousands of fruit so that they could test the cold tolerance of Queensland fruit fly and apples and kiwi fruit, so that they could generate enough data to look at the lethal times, various lethal times. We've just chosen 99% mortality here on apples, jazz apples, green kiwi fruit and gold kiwi fruit. So this added to our cold data package. You can see again that the third instars tended to be more tolerant, and we could take the kiwi fruit data out of our study and compare it with previous studies on kiwi fruit. So here we've got our data on Q-fly. We're comparing it with some Western Australia data on medfly at lower temperatures, and we can see that medfly is going to be a whole lot more tolerant than Q-fly on at least kiwi fruit. The next steps, well, we're planning on conducting further studies on kiwi fruit and apples for medfly at 3-degree cold treatment studies. Melon fly is such a poor host, it's going to be a real challenge to do that one. And oriental fruit fly, we've got evidence to suggest that this is going to be the least tolerant of the species, but I guess is it going to be enough evidence to convince regulators? So the outputs from the project, this review is being used by New Zealand government, Australian government and the quarantine researchers. It's a great resource. The prioritisation of market access research for each major industry has been really valuable to them. The provision that we've provided efficacy data for those shorter combination treatments for air freight. There's a cold data package that's been developed that potentially can be a repository for cold treatment data so we can compare across a range of studies. We've got further information on the host status of apples and kiwi fruit to fruit fly, and we've begun the process by looking at the cold mortality data on Queensland for Queensland fruit fly on apples and kiwi fruit. It's been a blast. This is the end user perspective. Basically, you can read that because I've just had my 2 dings, and we'll leave it at that, shall we?